So, hi, everybody. Thank you for being here. I'm really excited to introduce Naglis Subačius, who I've been able to work with now for 2 years. It's been really awesome to be able to work with and all the things that you took on. Naglis came by way of University of Illinois, Champaign, or Urbana-Champaign, Bachelor's of Science in Natural Resources and Environmental Science, and had a lot of really good skills. And everybody was recommending him that he'd be able to kind of like grow and take on new things, but he hadn't had a lot of opportunity to do so. And you took on a really big opportunity to kind of like grow from experiment that was kind of tied in with some collaborators at the University of Minnesota and Cedar Creek, something that had been running for more than 10 years, and kind of like grow a symbiosis type of experiment that you were really interested in, by using some new skills that you hadn't been able to kind of like harness before. You leaned in on help from a lot of collaborators and a lot of colleagues, and definitely took it in your own direction, and kind of we learned some really cool things, and I'm excited for what he'll be able to share with you. And you did some awesome work. And after this, starting next month, he'll be doing a PhD program at the University of Colorado Boulder. So more great things coming. Yeah.
Cool. Thanks, Matt. Okay. Well, this will jump right in. Thank you all for being here. My name is Naglis, and my thesis was called Linking Rhizosphere Biodiversity with Drought Response in Trees. And so we'll start with drought. As you all know, the climate is changing, and as that occurs, one change we expect to see is that droughts will become more common and also more intense. Zooming into the Midwest specifically, we expect to see that although the total amount of rain might not decrease dramatically, it will fall in more intense storms that occur more infrequently. And so you might imagine there that instead of percolating deep into the soil, water could run off to streams instead, which might advantage trees with shallower roots, something like this. And so that's all to say that trees are diverse, and different trees will respond to different kinds of drought in different ways. Moreover, they're also interacting with each other in forests and plant communities. And so the short-term effects of drought, which affect how plants interact with one another, can lead to long-term drought legacies that last beyond the initial stress. And so one of the main goals of my study was to better understand this diversity component of how different trees respond to drought in order to better understand what forest will look like in the future. And so starting off above ground, how can plants respond to drought? This has been fairly well studied, largely because above ground plant parts are not buried in the soil, so you don't have to dig them up to look at them. And so there's a decent amount of literature here. We see things like changes in leaf shape, changes in stomatal conductance, so how much water and carbon dioxide is being exchanged with the atmosphere, as well as changes in osmolarity. So plants can invest carbon to increase the number of molecules behind their cell membranes in their leaves in order to draw water out of their vascular tissue and out of the soil. And so both of these two traits tie in with this idea of hydraulic strategy, where a plant can be isohydric, heavily regulating the water potential in its leaves, or it can be anisohydric, more of a water spending strategy, where it uses water more regularly, but might be more susceptible to drought. And so we don't see all of these responses in any one given tree at the same time. One way to consider why certain strategies show up in some trees and not others is this idea of a fast to slow trade-off in ecology. Very widespread idea. For our purposes, it might be better to term it as speed versus security. A plant can grow very quickly, acquire a lot of resources, be very competitive. However, that might come at the cost of being resistant to stress, such as drought. And so really the crux of my study is looking at these dynamics below ground, where things get even more complicated as we add another axis of diversity. So here, recent literature has described that not only do roots occur fast to slow, but also on this axis of DIY versus outsourcing, where in order to acquire resources, a plant might grow a very robust set of roots with lots of surface area to pull water and nutrients out of the soil, or it might instead invest that carbon into partners like mycorrhizal fungi to do it for the plant. But in that case, the plant's roots itself are a bit more reduced. And so this is kind of a recent finding that researchers have termed the root economic spectrum, and we don't quite understand how it works for across the diversity of plants. One more time, it's also important to understand that these effects occur in the context of plant communities, where plants are competing with one another, maybe facilitating each other, and might even share mycorrhizal partners. Another thing to note is that when this research was first coming out back in the early 2020s, late 2010s, there was kind of conflicting evidence on whether or not we see plasticity on this horizontal DIY, do-it-yourself outsourcing axis within a given plant species. Originally, the research focused more at the broad interspecific level. And so all of this talk about outsourcing, but who are plants outsourcing to? Well, first of all of this occurs in the rhizosphere, which is a microbiome immediately surrounding the roots of plants in the soil, fed by carbon exudates from the plant that really upregulate microbial activity there. And so the organisms that we'll be focusing on today are mycorrhizal fungi, arbuscular and ectomycorrhizal, as well as more generally bacteria. So starting with mycorrhizae, which have a big role to play when it comes to outsourcing. As I just said, we have two types. We have arbuscular mycorrhizas, which grow into the plant cells and form these coiling structures called arbuscules that are the site of nutrient exchange. And then we have ectomycorrhizas, which grow around the plant cells and exchange root nutrients at that cell membrane interface instead. And so given that these two symbiotes exchange nutrients in different ways, we have also come to understand that there's different root strategies associated based on your mycorrhizal type. For plants that outsource more to arbuscular mycorrhizas, they'll tend to have thick roots with a lot of cortex volume to host more arbuscules, whereas plants outsourcing to ectomycorrhizas instead need more surface area. So they have more highly branching roots with tiny, fine roots. Also, it has been shown that mycorrhizas can have a big role to play in alleviating drought stress for their host. This is more well established in arbuscular mycorrhizae and seemingly context dependent in ectomycorrhizas. And there's also ties with evolutionary history here. So the arbuscular symbiosis is something like 400 million years old. Generally, we don't expect to see that a plant hosting arbuscular mycorrhizae will swap to ecto or vice versa anytime soon. Okay, so that covers mycorrhizas, but we also have bacteria, and these occur in the rhizosphere based on kind of selection from the bulk soil. So the bulk soil actually contains a greater diversity of bacteria. However, the bacteria that do make it to the rhizosphere end up becoming more active because of that high abundance of carbon exudates. And so to a certain extent, the carbon exudates that are coming from the plant actually determine what bacteria are recruited into the rhizosphere. And they can provide benefits like plant growth promoting bacteria that even produce plant hormones, as well as antimicrobials. And there's also, of course, pathogens that come and try and cheat the plant for its carbon. And all of this activity in the rhizosphere also means that over time, more carbon is being fixed in the soil, and this can have effects on soil texture and soil quality. And so Zoe took some really cool staining pictures and found that we did have a pretty intense amount of colonization on all of our trees in our study. So coming to the objectives of our work, as I kind of already mentioned, we want to see how do forests respond to drought below ground. One of the reasons why there is a research gap here is that roots and soil microbes are just kind of difficult to study. Roots are very fragile. You can see a lot of fragments here, so you have to be very careful not to damage them as you work with them. And then microbes really have only recently become generally affordable to study with the advent of more affordable genomics techniques. And so one of our goals was to look below ground, see what's going on, and link that with the existing literature on above ground. And then understanding better how trees holistically respond to drought, scale this up to ecosystem effects and ask, what will forests look like in the future? And so for our study, we selected 4 trees with an eye for diversity. We have a range of different mycorrhizal hosts, 1 arbuscular host, red maple, and then 3 ectomycorrhizal hosts, paper birch, red oak, and bur oak. They also have a half and half split of successional roles. So paper birch is a pioneer species. Red maple comes in at more of an early to mid successional stage. And then red oak and bur oak are typically thought of as climax species with some nuance there. Namely, that bur oak is known to be quite drought tolerant, so much so that it can actually serve as a pioneer species in sandy soils where no other woody plants can grow. We also see that red maple is known to be very tolerant of water stress, both growing in bone dry soils to waterlogged soils. And then paper birch, conversely, is known to be quite drought sensitive. Red oak, in general, prefers a wetter climate, but it does take on a rather conservative growing strategy, so it can still tolerate stress. So speaking of growth, with our early successional species, we tend to see more rapid early growth as they try to outcompete their neighbors, whereas red oak and bur oak tend to grow slower, being better perhaps at tolerating stress, and they instead invest heavily in below ground robust roots. Going along with maple's ability to grow in diverse habitats, its roots have also been shown to be highly plastic, whereas paper birch is unique because it's our only species in the study that does not have a taproot. So because it has this fast growth lifestyle with highly acquisitive roots, however, they're also perhaps cheaply built. And then we see in red oak and bur oak, heavy investment in a robust root system below ground. And then in terms of hydraulic strategy, that water use we were talking about earlier, once again, red maple can grow in many different types of stressful environments. And so part of the reason why it's able to do that is it does have this heavy control over how it uses water. Whereas our other three species are known as anisohydric, so they take more of that water spending strategy. That being said, paper birch, the literature is a little bit more mixed here. There have been some studies proposing that it takes an anisohydric strategy, but all of this is really contextual based on climate, competition, and things like this. Okay, so to answer our questions, we planted a common garden with these 4 species. And if you don't know what that is, basically we planted all of our plants, our trees, in a greenhouse under common conditions and just manipulated a few things. And so this allowed us to get closer to a cause and effect type of situation where if we see a response in our drought manipulation, but not our water manipulation, we can pretty confidently say that water was the driving factor there. And so here we see our greenhouse about 1 month into the growing season. The plants were still relatively small, so they didn't need that much water at this size, so we didn't see too much drought stress yet. But by the end of the growing season, we definitely did. And we had a couple of hypotheses as to why this occurred. First of all, we believed that the symbiotic dynamics of plant roots and their rhizosphere microbes would shift with drought. So not only might the rhizosphere become less active, but also the types of interactions going on would be different. We also expected these changes would differ between, say, oaks, maples, and birches. And we also suspected that the strength of the mycorrhizal symbiosis might not be directly tied to overall downshifts in rhizosphere activity because they're so important for drought activity of some plants. Now moving on to roots, perhaps rather uncontroversially, we suspected that our roots would change with drought. But we did expect to see different changes in different trees. And really, there hasn't been a crazy amount of research here. So just documenting this diversity is useful. We also expected these shifts to occur along the root economic spectrum. And we expected feedbacks between rhizosphere community and root structure, where different root shapes would garner different microbes, and vice versa, different microbes might induce different adaptations in the roots. In terms of broad level experimental design, I'll make one note that Matt kind of touched on already, which is that we designed our common garden study to be complementary with the Forest and Biodiversity 2 platform up at Cedar Creek. So this is kind of a natural field experiment where they're looking at the effect of biodiversity and forests. And so they have 12 species, including our 4, planted in different mixtures in order to see what happens when different trees grow together. And so hopefully our more manipulated common garden results will be complementary to the work going on up at Fab 2, where it's a bit harder to induce these heavy manipulations out in nature. And so to that end, we started with bare root trees, just as they did when they planted Fab 2. Matt was there. This also gave us a controlled starting point. So we had a higher recruitment rate than we would have seen with seedlings. We also used a soil that mimicked Fab 2. I'll get onto that in a second. And we had two primary manipulations, which were the amount of water in our pots. And we also supplemented some of our pots with inoculum from Fab 2 to introduce similar rhizosphere communities. So a little bit more on the soil. First, we sterilized the bulk of the soil that we used to plant our trees using a steam sterilizer, so a heat treatment. And then we also collected inoculum up at Fab 2 to introduce those microbes. And so one note here is that we use con-specific inoculum, meaning that a maple in our common garden received inoculum from a maple up at Fab 2. And then by the end of our growing season, if you haven't spent a lot of time looking at roots, this might mean nothing to you. But we did see a pretty highly active rhizosphere with a lot of mycorrhizal mycelium as well. So a little bit more on the pot setup. We had that soil mimicking the type of soil we see up at Fab 2. So it was roughly 60% sand and 40% topsoil. And then we had that inoculum making up 5% of our soil. In the case that a plant was not inoculated, we still used a placebo of soil from Fab 2. It was just sterilized prior to planting. And then we planted at different densities. So we have monocultures, pots with one tree in them. Here you can see what a bare root tree looked like before the growing season. And then we have bicultures where we had two trees in a single pot. And here you can see Matt uprooting two trees at the end of the growing season. The roots really grew together to be intermingling. A little bit more on the specific manipulations we used. We had a full factorial design. So every possible combination of trees and different plantings, different species, as well as different manipulations, water and inoculation were represented here. First of all, we had our well-watered manipulation or drought manipulation. Half of our trees received ample water and half of them were water limited. We also had an inoculation manipulation. Roughly 2/3 of our trees received inoculum. The remaining third was left as sort of a control treatment. And then, as I said, we had this full factorial combination of our diverse tree species. In terms of the data we collected, we looked at traits above ground, below ground, and also within the microbial community. Looking above ground, we largely wanted to estimate growth rates, and we did this through the use of allometry. If you've never heard of allometry, think of like Da Vinci's Vitruvian Man, where your wingspan is equal to your height. Kind of the same thing with plants, where if you know one measurement, you can pretty accurately estimate another one. Here's a very simple example where if you know that the longest leaf on the plant is 10 centimeters, you can maybe estimate its height is double that. Our models were definitely more complicated than this, but this kind of gives you the general idea. And so we used allometry twice, once at the start of the growing season and once at the end of the growing season to see how much mass our trees packed on throughout our experiment. And this was important because we were using bare root trees and we didn't want to measure their starting point in our experiments. We wanted to measure what happened while they were planted in our common garden. And so for growth rate, we had two measures. First of all, absolute growth rate, the total amount of growth throughout the season, and also relative growth rate, which is adjusted by the size of that plant, because a larger plant inherently has more access to photosynthetic tissue and root tissue, so it can grow faster. So in terms of what that means, here we have two plants that doubled over the course of the growing season. The top one packed on more mass relative to its size, so it had a higher relative growth rate, whereas the bottom one packed on more overall mass. We use this measure to quantify success during the growing season in three ways. First of all, just the ability to grow more, then the ability to grow consistently across manipulations, and also to avoid dieback. So here we can see the top plant did quite well across conditions. It grew roughly the same, whereas the bottom plant grew well in well-watered conditions, but did not do so well in drought. Lastly, above ground, we also measured osmolarity, which I already explained a bit earlier. To do this, we took our plants, dark adapted them to turn off or to deactivate the photosynthetic machinery. And then I came in after they spent the night becoming fully hydrated and wearing a green headlamp, collected leaves and ran them through a osmometer. And so this allowed us to estimate the concentration of osmolytes in the leaves, which tells us a little bit about their turgor loss point, or how well could they withstand wilting in the face of low water availability. For roots, we had one workflow, but it was relatively involved. We removed our plants from their pots gently as to not damage the root structure, washed off the soil, and then collected 3 subsamples of their fine root growth. Once again, these are bare root trees, but we were only interested in the growth that occurred during our growing season, so we only looked at new growth. And then we edited these images digitally to maximize contrast so that we could use them in a software called Rhizovision, which allows us to digitally collect measurements on these plants. So you can see here an example root that becomes digitalized, and we get a whole range of measurements. We didn't use all of them. I'll explain the important ones in a second here. Rhizovision is useful. It's free. There are competitors that cost thousands of dollars, but in some ways, Rhizovision has been shown to be more accurate than them. It also allowed us a standardized workflow. So taking root measurements can take a long time, and using Rhizovision allowed us to measure a lot of roots. And we did see a trade-off here, though, where we got a lot of data, but we did not take some detailed measurements that you would see elsewhere in the root literature, such as specific root length. So one of the key features of Rhizovision is you can differentiate by diameter and take traits for different diameter ranges, which is important when we consider that fine roots are the acquisitive tissue of the root system and coarse roots transport material. So here we see these skinny roots here have a high surface area to volume ratio. They have a lot of contact with the soil, and so they're good at grabbing up water and nutrients. And then they transport that into the thicker roots, this green one here, that then takes that water and moves it into the rest of the plant. But these coarse roots are generally more lignified and woody, so they're actually not so good at acquiring resources on their own. In terms of the traits we looked at, we measured diameter, number of tips per length. You might remember that these have some tie-in with outsourcing to arbuscular and ectomycorrhizas. And then we also looked at some more kind of abstract measurements, like the surface area to volume ratio of the fine roots. How efficient were they at acquiring resources, as well as the surface area of the fine roots corrected by the length of the coarse roots. So how much acquisitive tissue was feeding the transport tissue. That's more of a measurement of, say, acquisitive magnitude. And then we also looked at soil diversity. Initially, we planned to look at 3 dimensions of diversity, namely the bacterial component using a 16S primer, the fungal component in 2 ways, one using the broadband ITS primer, very common in the literature, and we also tried to use a primer set specifically targeting arbuscular mycorrhizas, which are notoriously difficult to sequence, and we unfortunately failed to sequence them. So this is the last you'll be hearing of that today, pretty much. In order to collect our DNA, we separated the rhizosphere from the soil, basically using agitation with a vortexer and then a couple of centrifugation steps. And then we used a commercial soil DNA extraction kit to remove the DNA from our soil. And then we did a 2-step PCR prep before sending off our DNA to be sequenced at the University of Minnesota's Genomics Center. And so once we got our data back, we had to process it before we could use it. And to do that, we used this pipeline where we removed unwanted reads or unwanted parts of the reads. And then we used a tool called Dataset to basically bin our reads into different amplicon sequencing variants. I'll explain that in a second. And then we assigned taxonomy there to using publicly available databases, and then did analysis in R using tools like phyloseq, amongst others. So when we got this data cleaned up, what we had were, for each given microbiome sample from our different trees, all the different ASVs we detected there. ASVs are basically like your operational taxonomical unit. After Dataset, we were confident enough to say that this ASV came from this organism and this one came from this organism. They came from different organisms. And then we also had their relative abundance. And this is important to note. We did not have absolute abundance, so we cannot say that organism 1 was 10 times more abundant here than in another sample, but we can say that proportional to the other microbes we sequenced, it had a higher relative abundance. And so with this data, we looked at alpha diversity, both the total number of species and also their evenness. And then beta diversity using ordination techniques, and community composition using differential abundance analysis. For the results you're seeing today, I use a tool called ANCOM-BC, but I'm still not settled on what the best one would be. This was just a little bit easier to use for now. In terms of results, you can't really see the tags, but we have, as I talk about them, we have our microbiome results, our below ground results in brown, and our above ground results in green. So overall, what patterns did we see? First of all, we found that our two stressors, competition and drought stress, were impactful in interacting ways. So here we have relative growth rate plotted in our bicultures and our monocultures, and we see that the effects of drought were really only pronounced under the effects of plant competition. Looking at the profiles of our different species, here now we have absolute growth rate by all of our species. We see overall, birch grew the most, then Acer, and then both of the oaks were relatively comparable. However, we did see some differences by water treatment, where birch took quite a large hit in its mean growth rate. However, it still grew the most among all of our species. Acer took a little bit less of a hit and still outgrew Quercus rubra, although not Quercus macrocarpa. Both of the oaks had pretty similar growth rates regardless of water treatment, although it seems like macrocarpa was maybe a little bit more efficient at using water. So here, this data alone might be a little bit misleading. We see paper birch grew more than all of the other species no matter what, but it did pay a cost where we saw higher leaf dieback as well as stem dieback, not pictured here, under drought for paper birch specifically. Keep an eye on that. We'll return to the dieback idea in a moment here. But finishing out relative growth rates, we saw that inoculation had an effect as well, where maple had a higher growth rate with inoculation, and red oak actually decreased its growth rate with inoculation. And then although inoculation itself did not have a simple effect on bur oak's growth rate, we do see that there was a relationship where as fungal diversity in its rhizosphere increased, we saw a positive correlation with relative growth rate, and there was no such relationship in the other species. We'll return to that in a second. Now, though, looking at osmolarity, we see universally across all of our species an upregulation in osmolarity under drought. However, the size of this response did not differ between species. What did differ between species was their fundamental base osmolarity level. And so we see here, Acer rubrum, compared to the oaks, had a relatively low osmolarity level, although I wouldn't say 600 is low necessarily. But this does follow in with that idea that Acer is an isohydric species, which can regulate its water use. And so perhaps osmolarity is a smaller part of its hydraulic strategy. Whereas we see Quercus, two of our anisohydric species had a high osmolarity level, which makes sense given that they take this water spending strategy, but are also slow growers, that they invest in osmolarity that can safely back up that water spending strategy. Curiously, paper birch, ostensibly maybe an anisohydric species, had a very low osmolarity among our species. We also see an interesting relationship when we compare osmolarity versus growth rate, where for these three species up here, not including birch, there was a trade-off where the plants could either invest carbon in growth or osmolarity, but not both, which makes sense. You can't have your cake and eat it too. But at first glance, it almost seems like paper birch does. And to understand this, we have to remember a couple things, which were that paper birch grew the most. Being a larger plant, it required more water. And in order to grow more, it seems that it invested less in stress-tolerant traits, such as osmolarity. And we also saw more dieback overall. So it seems that osmolarity regulation was important for the drought response of birch, but overall, it was not able to tolerate stress as well as their other three species. So just wrapping up our above ground trends, all of what I said helps to paint the picture of paper birch being a pretty fast growing species with relatively low stress tolerance. Acer also grows pretty fast, but it was able to downregulate and tolerate the drought stress in our study. And then both of the oaks grew quite slowly. And it's important to note that their smaller size means that overall they required less water. So relative to the birch, the effect of our drought treatment was a little bit weaker on the oaks. They were able to avoid that drought stress. And then we also saw those relative growth rate outsourcing trends in these two species. So just kind of painting a picture here of our different species profiles, we see that Acer rubrum is risk tolerant. It can grow well while handling stress, whereas Quercus, being a more conservative species, kind of avoids that stress instead of trying to lean into it. Whereas paper birch kind of puts all of its chips into growing well when water is available. And we see that it was a very competitive species there, but this gamble did not pay off under drought when water was not available. It kind of paid the consequences. Moving on to our below ground organs, we see that Acer's roots were pretty thick and also sparse in terms of branching intensity, which contrasts what we saw in Quercus, where the roots were finer with more branching intensity. You can see all these little root tips up here that are not present as much on our maple. And also these two images are relatively to scale. And so this picture lines up with our idea of an outsourcing arbuscular species, needing those high diameter roots to host arbuscules, versus an outsourcing ectomycorrhizae species, needing that high surface area to host more ectomycorrhizas. It seems that paper birch was kind of an intermediate, where it did have more root branching than Acer and also finer roots. However, these branches were a little bit, these first order tips were a little bit longer than what we see in Quercus. And overall, it produced more acquisitive tissue, so more of a DIY response in general. And so all of those patterns I just set up held true when we looked at these roots in a principal components analysis. So if you don't know what this is, basically, we took all of our root traits and squashed them down into 2 dimensions to see what drives variation in our species. And we see that there's some broad patterns by morphology and architecture. Morphology and architecture show up on both of the principal components, but it is more dominant in principal component 1 that morphology is more dominant in principal component 1. We see what does the average length of root look like? How thick is it versus how much? What is its surface area to volume ratio? Looking at principal component 2, we see more patterns of architecture, such as how are the roots organized? How much branching intensity is there? And so this brings me to some recent publications in the root economics space literature, suggesting that we can't use the same traits to quantify outsourcing in arbuscular mycorrhizas or in ectomycorrhizas. The original publication back in 2020 that came out with this outsourcing idea suggested the use of diameter to quantify it. But we see that the effect or the importance of diameter for arbuscular species outsourcing is much greater than it is for ectomycorrhizas. And so this paper suggested using branching intensity instead to quantify outsourcing for ECM species. And it appears that we did capture both axes of outsourcing diversity here, as well as the DIY strategy with greater acquisitive magnitude produced. Okay, so here's the same PCA, just split up by our different species and our water treatment, as well as neighbor manipulations. You'll see that the axes are a little bit different, but this is all the same data from the last slide, and we have the loadings up here to help you see what you're looking at. First of all, we see that bur oak was actually not very responsive to water stress in its roots. There's a large amount of overlap here with just a few points escaping. Whereas a species like birch seemingly was quite responsive, where under drought in red, it had these more acquisitive DIY roots with greater acquisitive magnitude, whereas under well water conditions, it had more branching and higher diameter roots, which would be indicative of an outsourcing strategy. It also seems that this had something to do with neighbor interactions, where these high branching roots also co-occurred when birch was growing with Quercus macrocarpa. So for now, I'll just say who you're growing with does matter, and we detected quite a few complicated neighbor interactions. We have enough to talk about today with just the drought stuff, so I won't dive into that too much more. I will note, though, that it seems like these roots are becoming less acquisitive, right? They're growing bigger in diameter. However, this was not associated with a decrease in growth rate. So it seems that this is, at least in some part, associated with more outsourcing. So I won't dive too deeply into here, but basically all the patterns I just described were not only based on visualization. We also looked at our traits more rigorously. Here we have average diameter. For birch, we see it was higher when growing with Quercus macrocarpa, and we see similar trends in the other ectomycorrhizae species. We also see that paper birch did have trends of more DIY style roots, especially under drought conditions where it upregulated its fine root surface area to volume ratio, whereas this relationship did not hold true in the other trees. Okay. We did detect an interesting trend when looking at how growth rate was related with root traits in Acer, though. So that's this blue line here. We see as it increased its branching intensity, what you might call an acquisitive trait, its growth rate actually went down. And inversely, as its average root diameter went up, also thicker roots, less acquisitive, its growth rate went up, which does fall in line with this idea of an AM outsourcing species needing higher root diameter to host more arbustules and performing better in that context. Okay, so wrapping up our root trends, we see that our streamlined method of digital root phenotyping seemingly did capture multiple dimensions of diversity, both AM outsourcing, EM outsourcing, and a DIY strategy. Now, this publication mentions that given how different these strategies can look across species, that we need to take a modular approach where the methods used to quantify root economics is specifically tailored to the species being studied. And it seems that our high throughput, high sample size method here was effective in capturing intraspecific variation more granularly than other broader spectrum species have in the past. Now, looking at trends of DIY versus outsourcing, we see that it appears paper birch traversed the DIY outsourcing spectrum pretty intensely with drought. Under well-water conditions, it had this high branching outsourcing type of lifestyle, whereas under drought conditions, it had more of a do-it-yourself lifestyle. And this makes sense when we consider that ectomycorrhiza specifically can be pretty sensitive to reductions in photosynthesis. And we saw that paper birch had a lot of leaf dieback, so it was not able to fix as much carbon. Potentially, under drought, paper birch was no longer able to afford this symbiosis. We also see that Acer's relative growth rate aligned with the do-it-yourself outsourcing axis. Potentially, this could be more pronounced in arbuscular species because their roots are inherently less robust, less acquisitive. Perhaps without a partner, they're not able to grow as well. That being said, we did not measure root hairs, which have been shown to be important for DIY response in species like Acer rubrum. And also these results definitely would have been enhanced if the arbuscular sequencing worked out, fortunately. And then one more time, Quercus, the roots were pretty consistent under drought and well-water conditions, specifically for oak. So one more thing, one more side of the data is tying in the microbes. So here we have alpha diversity, and it almost looks like I'm showing you the same plot 3 or 4 times. And so what we have is our fungal diversity, total number of species, and evenness, Shannon diversity, as well as the same thing for bacteria. And we see a consistent trend where our fast species, birch, has less diversity than our more conservative species, maple and red oak, sometimes bur oak. We also saw that diversity in our microbiomes was responsive to manipulation. There were simple effects where here bacterial diversity was increased with well-watered conditions. We saw similar effects with the fungi, but they were more complicated, perhaps better visualized with a principal components analysis. So just to help you see what you're looking at, here, every single point represents a single microbiome sample from one plant, and points are spaced based on how similar their microbiomes were. So this microbiome was very dissimilar from this microbiome, whereas these two were potentially more similar, were more similar. And we see that drought treatment does drive changes in our microbiome structure, and it depends on what tree species the microbiome is growing upon. Here we're looking at the fungal data, and it does seem like there was a weaker effect on microbiome with inoculation as well. Moving on to bacteria, we see strong clustering in the uninoculated condition. However, moving forward, I'm really more interested to talk about the inoculated microbiomes. So here we have just the inoculated data split up by water treatment, and we see specifically in birch some strong clustering for the well-watered microbiomes and the drought microbiomes. It seems like there was a divergent response here based on water treatment in the birch rhizospheres. Not so much in the other 3 species. Okay, that was a lot of data, and I hate to tell you, we still have one more pretty dense slide. So I just wanted to give us a second to digest. Okay, they're doing pretty good on time. All right. So we see that our different manipulations do drive changes in community composition of the rhizospheres, but we wanted to see what was actually changing. And so this is where that differential abundance analysis comes in. We looked at how did microbes respond to our treatments. And so here we have a lot of volcano plots. And if you've never looked at one, I'll try and help you read it. First of all, we have our fungal species plotted on the top, bacteria on the bottom, split into columns based on what tree rhizosphere they were growing in. And so each individual point on these plots represents an individual species from the rhizosphere, averaged across Acer, birch, stuff like this. And they're color-coded based on how strongly they were associated with our well-water treatment in blue or our drought treatment in red. So species here on the right were many times more abundant under well-watered conditions than they were under drought. That's our log fold change, and the same holds true in reverse for the drought plants, or the drought microbes. And then on the y-axis, we have our adjusted p-values. So was this difference significant, or was it just noise? And so below this threshold, these more grayed out points, we did not find a significant difference there. So for today, I'll mostly focus on the fungal data. This bacterial stuff I only recently got working, but any of you microbiome people, if you recognize some names, be sure to shout it out. I know it's really small. Yeah, sorry. I'll point out that I'll name the microbes I'm talking about as I go, though. So in Acer, we found few microbes to be significantly turning over just with water treatment. That could be related to sampling power, though, so I don't want to draw any strong conclusions quite yet for Acer. But we do see some interesting trends in birch and Quercus macrocarpa, where in birch, under well-watered conditions, we see a bunch of ectomycorrhizas like Sphaerosporella, Pizziza, I think Hebeloma can form ectomycorrhizas, whereas in the drought condition, we see one genus pop up multiple times. And I'll spell it out for you. Paraphoma, 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 a couple more paraphomas, which is a known plant pathogen. So we see, well, I'll unpack that in a slide. For Quercus macrocarpa, though, we see the same pattern does not hold. Actually, under drought, we see scleroderma is strongly associated with drought condition, which is an ectomycorrhizal fungus. We don't see any on the well-water condition, though. One thing to note, though, is that I still haven't really looked into what species are hitting here below the significance threshold, so we can't draw holistic conclusions yet, but we definitely see some interesting patterns. So one more set of plots, this time split up with our root traits. So remember, we have principal component 1, the horizontal one represents morphology. Those are these two plots on the left. And then principal component 2 represents architecture, and that's these plots on the right. And we notice a couple of interesting trends where on principal component 1, as roots are more thick, we see ectomycorrhizas, which makes sense. As that fungal mantle enshrouds the root, it makes it thicker. Whereas as roots become skinny, we find those pathogens pop up again. They're strongly associated with roots over here on the DIY versus fine root side of the spectrum. With branching intensity, we actually see ectomycorrhizas on both sides, more of them here with branching intensity. However, we see the pathogens again pop up under this DIY side of the principal component. Okay. Also, all of these results were independent of what species the microbes were growing on. So this is actually a pretty interesting finding. Not many studies have looked at the relationship between root traits on their own and microbes. So what does this all mean? First of all, we see divergent trends in paper birch, where under well-watered conditions, it seems to have strong ties with ectomycorrhizas. This also seemingly has something to do with neighbors, but that remains to be unpacked. Under drought, remember, it shifted towards a more DIY root space, and at the same time, it seemingly was more vulnerable to pathogens. One way we can understand this is by thinking about the idea of a growth defense trade-off. Some studies, or one study, found that for roots specifically, fine roots are often the site of infection by pathogens. Specifically, finer roots, more acquisitive roots, are actually more susceptible to infection. So we see that paper birch under drought did move in this direction of more acquisitive, cheap, fine roots, and it was infected more by pathogens. There are other ways to explain this, but this nevertheless could be a part of the bigger picture. For our conservative roots, namely these two species, remember we saw higher fungal diversity, and now is a good time to consider that microbes also exist fast to slow. And so recent research has shown that conservative roots tend to garner conservative microbes. And this is because they put out more recalcitrant carbon exudates that are more difficult to assimilate for the bacteria. And so these conservative microbes tend to grow more efficiently and actually assimilate more of that carbon into biomass that stays in the soil rather than respiring out of the soil. Specifically, it ends up coming into the soil, remaining in the soil through the fungal part of the trophic pyramid. So perhaps this is related with our higher fungal diversity. We don't want to conflate diversity with function though, so there's still more work to do to see whether or not this relationship was real in our study. And then lastly, we see that Quercus macrocarpa had distinct mycorrhizal ties under drought, which we actually did expect, given that previous studies have shown that because mycorrhizas are so helpful with drought, that they can be more abundant, or relatively more abundant, under those stressful conditions for the plant. However, kind of going against those findings, we did not see the same trend held for other species like birch and potentially red oak as well. And then lastly, you might have noticed I haven't been talking about red oak that much. That's because it had some pretty complicated things going on with pathogens and neighbor and dieback that we're still unpacking. We've got enough to chew on. So one last characterization of our species, we see Quercus macrocarpa was a slow grower that seemed to have strong ties with mycorrhizas. Paper birch was a fast grower, although its outsourcing versus DIY strategy was seemingly context dependent, both based on water treatment and also neighbor. Acer, on the other hand, did have these more reduced outsourcing roots, although it also grew faster than both of our Quercus species. And then Quercus rubra grew the slowest and had some neighbor interactions. It being an ECM species will put it closer on the DIY side of the spectrum than Acer. Okay, so what does this all mean? First of all, our results show that drought does drive roots along the root economic spectrum. However, these changes are not linear. This is perhaps best shown with birch, where with well-watered conditions, it moves horizontally towards the outsourcing side of the spectrum, whereas with drought, it moves diagonally. One thing to consider here is that root strategy is not independent of the rest of the plant. Truly, these results would have been more difficult to understand if we were not looking at things like osmolarity and leaf dieback. There's also something to consider here that plasticity of plant traits is a heritable trait. So understanding why certain plants might be more capable of traversing the root economic spectrum could be important for understanding their success under drought. We also found that microbes were related with root traits. First of all, mycorrhizal type seems to matter. Remember that arbuscular species tend to be more on the outsourcing side of the spectrum with more reduced roots, and ectomycorrhizal species have inherently more branching acquisitive roots even without their partner. And so moving in this DIY direction might have different outcomes for these different host types on average, where without its mycorrhizal partner, an arbuscular host might be less able to grow, have lower production overall, whereas ectomycorrhizal species might be more susceptible to pathogens given their finer root structure, and also because they're losing the antimicrobial properties of ectomycorrhizas. Now, these are just two trends that we saw in our data with two species that do have other explanations, even within the context of our data. So these are certainly not conclusive findings, but definitely interesting directions for future research. We also found, or other studies have shown, that carbon cycling occurs on the fast to slow root economic spectrum below ground. And so we found that drought drives root economics in nonlinear ways, right? So this means that understanding when roots might move in the fast direction versus the DIY versus outsourcing direction will be important for understanding overall carbon cycling dynamics in the future, which has implications for greenhouse gases and global warming. And then lastly, what does this all mean for forest assembly? Well, we found that root drought response is nonlinear, so more work is left to be done to describe when, where, and why different plant species respond to different kinds of drought in different ways. And it also seems that mycorrhizal type really matters. And this is important to consider in the context of human land use. It's been shown that as we clear cut forests and plant crops and till the soil, we generally decrease the amount of ectomycorrhizal partners available in the soil. And so this has implications for things like reforesting as we try to introduce ectomycorrhizal species back into these places. And also they can help explain why we see such a patchy arrangement of trees and forests in places like the eastern US, where these forests are secondary forests previously used for agriculture. So overall, our results indicate a need for more research to understand better how different types of root economics, different types of drought, and different mycorrhizal types all interact in order to better predict how forests will look in the coming decades. And that's it. Yeah, I'd like to thank a lot of people and a lot of people who gave me money. Of course, we have MSU and also the Faculty Research Grant, the Dirksen Peterson Research Fellowship. Our microbial work really would not have been as comprehensive without it. Also funding through the Radical Herbarium and funding and help through the Rise Bio program. I'd like to thank my committee who know a lot more about working with plants and microbes than me. Matt, Dr. Naker, Peter, other people who also helped me in an advisory way. Alex, Jake, Luke, Kathleen, graduate students who also helped get the study going, April and Fen, namely. I remember you guys helping me to shovel kilos of sand all day. And then also help from a lot of undergrads. That's it. Thank you, guys. Yeah.
I know how small of an area there is to work in those greenhouse bays on campus. I was wondering if you noticed that there could have been any canopy crowding, even the monocultures in soil space that could impact the above ground.
Yeah, that definitely was something that occurred. And our study was planted at a finer density than you would see out in nature. That being said, they actually saw similar things at Fab 1, where our study was designed to mimic. Their original planting, Fab 1, was so dense that they realized, like, hey, this doesn't really mimic what we see in forests. So Fab 2 was planted to be more widespread. And I would say the canopy effects probably did have a role in our study, but given that we were looking at competition, perhaps that's not the worst thing. And we can look to Fab 2 to see what does a more natural ecosystem look like.
That just overall had showed like they were more resistant to drought than other species.
Wait, hold on. People online want the question repeated. So
okay.
The question was there any bicultures that were more resistant to drought
than either species up?
Yeah, than monocultures or species by themselves.
Yeah. I don't think we detected any that were more resistant. I think we might have detected some dynamics with common mycorrhizal networks where we did see increased growth in some of the mycorrhizal partnerships. Unfortunately, those p-values were like 0.051. So I didn't talk about it today. We did see some combinations that were more susceptible to drought, though. Specifically, birch and red oak growing together had a higher prevalence of pathogens that I don't really know why, but they died a lot more than any other combination. Yeah.
That's why muscular were more difficult to see.
Well,
wait, repeating question.
Oh, sure.
Yeah, the question was why are muscular mycorrhizas were more difficult to characterize?
Yeah. So the primer set we used I believe, was originally designed for qPCR. So it's highly specific in that it will only target arbustular mycorrhizal fungi, whereas the IPS primer is kind of more broadband. That's generally what you use to sequence all fungi. Glomeromycota are weird because they have so many different number of nuclei. And also, I think because we used this more specific primer, it also had a lower ability to pick up any DNA in our samples. Also, Glomeromycota is way less abundant than bacteria. And I had one more point. Also, we would expect that the biomass of AM fungi is largely held within the plant root, not on the outside in the rhizosphere, which does not hold true for ectomycorrhizas. And so I think if we wanted to better sequence them, we might have used the plant root tissue as well. Yeah.
2 questions online. I'll read them. 2 questions online. First one, noticed in your methods that you sterilize the soil to start with for a clean slate in the microbiome. Curious why you didn't do that to the bare root trees you planted.
Yeah, that's definitely something I've been thinking about a lot. I think within the scope of our study, sterilizing the roots would have been interesting in an ideal way, like if we had that blank slate, but it is kind of impossible to get a blank slate. Namely, I mean, we were interested in the root microbe feedback, right? And so if microbes are important in how an early root system develops, then nuking those microbes before the start of our study might have made our results less applicable. And also, similarly, sterilizing the roots almost certainly would have had an impact on how the plants grew, which means that our results would have been less comparable with Fab2.
Then a second question, can you speak on how you chose the roots you analyzed, given your saplings already had roots?
Yeah. Well, like I mentioned, we only looked at new growth. And so as plant roots develop, they start at that root apical meristem and then slowly over time spread out more distally into the soil. So we started at the first order roots, or the most distal roots, which were guaranteed to be new growth. And then we followed the root system back to the transportive tissue, usually either the 4th or 5th order, depending on species, and then cut there and took measurements on the new fine root growth. Yeah.
Or in your next step in life, you're going to continue working with Rhizosphere research.
Yes. Yeah, so my next study has already been determined. It's NSF funded, which is cool. Basically, what we're looking at is that there's been a lot of shrub encroachment of willows into alpine tundras at this LTER, Niwot Ridge. And they've been trying to predict why it happens using stuff like, north-south facing slopes and snow melt, where it tends to accumulate in lower soils. But they haven't been successful. It seems to be really unpredictable. One reason they think, or one hypothesis they have now, is that Salix willows are a dual host fungus. And so potentially its success in invading these herbaceous areas depends on the availability of ectomycorrhizal hosts. So that could have something to do with like density dependence, where new willows only grow near other willows, even though they have very easily dispersing seeds, stuff like that. Yeah, I agree. Yeah.
I have some questions, but one thing that I'm sure is just like on your list to look at eventually, but like how much overlap is there in the microbial communities between your different tree species, right? Like strictly from like a presence absence perspective.
Yeah.
Right. Like you showed these volcano plots, but like some of those things might be like only in the, red oak or whatever.
Yes. Oh God, I animated these too much. Yeah. It seems that, we have a decent amount of overlap in terms of membership. These are Bray-Curtis distance matrices, but they were almost identical to the Jacquard ones. So based on community membership. Yeah, I haven't explicitly looked at that yet. I will say with mycorrhizas, it's known that early successional trees tend to find early successional mycorrhizas. At least in nature, I mean, our greenhouse is different. So that might have been a role to play when it comes to why certain fungal diversity was different in one place than another.
Just, thinking about things like that, that pathogen chance. And
Yeah.
things like that, right? It's like, was that there? in the
Yes,
first one?
that's a good point. I guess we did look at it a little. The paraphoma was found across all treatments, but it was really abundant in just a few. That's the one at the top here. Yeah.
That's cool.
Yeah. Oh, yeah, we did. Today's results have either been only from inoculated plants or controlling for the effect of inoculation, so kind of reflecting as much as we can what we'd see in a natural ecosystem. That's definitely like one of my next steps, though, to see what was actually coming in from the original source.
All right, I think we'll wrap up there. There's no more questions online, but yeah, thank you very much.
Cool. Thank you, guys.